Welcome, curious minds, to another deep dive. Have you ever truly stopped to consider what a virus is?
It's a good question.
Beyond just a tiny germ, that is. We talk about them constantly, yet their fundamental nature is actually, well, one of the most bizarre and paradoxical things in all of biology.
It really is fascinating stuff.
For instance, did you know that viruses aren't even considered alive in the traditional sense? And yet they hijack living cells with incredible precision, literally turning our own cellular machinery against us just to reproduce.
It's a profound, almost unbelievable concept. They exist right on that enigmatic boundary,
Yeah.
blurring the lines between what we define as chemistry and what we define as life.
It's one of the most fascinating subjects to explore.
Absolutely.
And today we're embarking on a deep dive into this fascinating, microscopic, and often misunderstood world of viruses. We've been sent a stack of intriguing sources, from detailed scientific texts on viral properties and replication to a really insightful look into how these enigmatic agents are studied in labs.
Yeah, some really great material. Our mission today is essentially to unpack these sources for you. We aim to give you a shortcut, sort of, to understanding the fundamental nature of viruses.
Right.
How they manage to survive and reproduce by, well, basically taking over host cells, and the ingenious methods scientists have developed to cultivate and identify them.
We're going to explore their bizarre structures, their incredibly clever survival strategies, and the sometimes, frankly, alarming ways they interact with the cells they infect.
You're in for some genuine aha moments, I think.
Definitely, because the more you learn about viruses, the more you realize how truly unique and impactful they are in our world. So let's unpack this.
Let's do it.
Let's start with the absolute foundation of understanding viruses. If you ask any virologist, the first thing they'll tell you is that viruses are obligate intracellular parasites.
That's the key phrase.
That phrase sounds very scientific, but for someone just trying to grasp the core concept, what does it really mean and why is it so foundational?
Okay, so that phrase, obligate intracellular parasites, it's the bedrock of viral biology, really, because it defines their very existence. It means they must live inside living host cells to survive and replicate.
Obligate meaning mandatory.
Exactly. They are entirely dependent. Unlike bacteria or fungi, which are independent organisms with their own metabolic machinery, viruses just cannot perform any of their metabolic functions or reproduce on their own.
So they lack the basic tools for life.
Pretty much. They essentially lack the tools for a self-sufficient existence. They completely rely on the host cell's machinery, its ribosomes, its enzymes, its energy supply, its basic building blocks to create new viral particles. This absolute reliance is what makes them so profoundly different from all other forms of life we typically think about.
So if bacteria are like independent, tiny, single-celled organisms with their own factories, then viruses are the ultimate squatters.
That's a good analogy.
They don't even work their own furniture. They just move into your house and use all your stuff to build copies of themselves. It's a profound distinction, really.
It is.
And this dependency also highlights why they don't fit neatly into the typical definition of a cell or unicellular microorganism, right? They're just different.
Precisely. They lack the complex cellular organization of bacteria. They don't have a nucleus or organelles, nor do they possess a true cell wall like bacteria do.
Okay.
And a really defining characteristic is that they contain only one type of nucleic acid, either DNA or RNA, never both.
Never both. That's huge.
Yes, and it's a sharp contrast to all cellular life forms, which contain both DNA and RNA. Furthermore, they completely lack the enzymes required for their own protein and nucleic acid synthesis. This isn't just a minor difference, it's fundamental.
Total reliance.
Totally. And instead of dividing through simple binary fission, like bacteria,
Like simple splitting in two.
right? Viruses multiply by a highly complex assembly process where their various components are synthesized separately within the host cell and then meticulously put together.
Here's where this really hits home, and it's a critical aha moment for many people, I think. This fundamental difference, this absolute reliance on the host cell's machinery and their lack of independent metabolism, is why antibiotics, which are designed to target bacterial processes like cell wall synthesis or bacterial ribosomes, are completely ineffective against viruses.
Exactly. No target.
This isn't just a fun fact. It's the core reason why treating a common cold, which is viral, is so different from treating a bacterial infection like strep throat. You can't just take an antibiotic for the flu, and now you know why.
That's exactly right. The mechanisms by which antibiotics work simply don't have a corresponding target in a virus. It's like trying to use a key for the wrong lock.
Makes perfect sense.
And another fascinating aspect, intrinsically linked to their unique nature, is their size. Viruses are profoundly smaller than bacteria, which was actually a key discovery in understanding them.
How small are we talking? Can you help us picture the scale here? It's hard to imagine something that small.
It really is.
Yeah.
Their minuscule size was actually the very first clue to their unique identity. Early scientists, trying to isolate the cause of certain diseases, discovered their filterability.
Filterability.
Yes, this was their ability to pass right through ceramic filters that were fine enough to retain even the smallest known bacteria.
Whoa.
This was a critical breakthrough that told scientists they were dealing with something fundamentally different from any microorganism they'd encountered before. They slipped right through.
So they literally slipped through the cracks that even bacteria couldn't? That's incredible. What's the general scale of that? Are we talking half the size of bacteria?
Are much smaller.
Yeah.
Generally. We measure virion size in nanometers.
Yeah.
Or mn, that's 1/1000th of a meter.
A billionth. Okay.
We vary widely, but to give you a sense, the tiny parvoviruses are only about 18 to 26 nanometers,
Which is?
making them barely larger than some very big protein molecules, almost molecular in scale. On the other end, the much larger poxviruses can be up to 300 nanometers. Now, to put that into perspective, a 300-millimeter poxvirus can be almost as large as the smallest bacteria.
Okay, so there's some overlap at the top end, but generally viruses are way smaller. Imagine something almost molecular in size, yet it possesses the incredible ability to infiltrate and wreak havoc on an entire living organism.
It's truly mind-boggling.
It is, and despite their microscopic nature and fundamental simplicity, viruses come in such a surprising variety of distinct shapes and structures. They're not all just little blobs, are they?
Not at all. While many animal viruses are roughly spherical or pleomorphic, meaning they can vary in shape, yeah. Others have very distinctive, almost geometric forms. You have viruses that are quite literally brick-shaped, like the pox viruses.
Oh, tiny bricks. Okay.
Then there are the striking bullet-shaped ones, such as the rabies virus, which belongs to the rhabdovirus family.
Bullet-shaped, that's memorable.
And even rod-shaped viruses, like the tobacco mosaic virus, which primarily infects plants, but was one of the first viruses ever discovered.
I'm picturing these tiny, bizarre architectural creations. And speaking of vivid imagery, let's not forget bacteriophages.
Yes, the phages.
These are viruses that specifically infect bacteria, and they often have these incredibly complex morphologies, often described as tadpole-shaped, with a distinct head and a tail-like structure, almost like little lunar landers.
Yeah, it's a very common description, yes.
Yeah.
It really helps visualize these microscopic entities when you think of them in such specific, almost mechanical forms.
It certainly does. And these shapes aren't just for show, are they? They're intrinsically linked to their function and how they interact with their host cells.
Absolutely. Form follows function, even at the nanoscale.
Okay, let's unpack this further and zoom in on the viral blueprint itself. We've talked about viruses being tiny and having distinct shapes, but what actually makes up a complete infectious viral particle? What are its essential building blocks?
Right, so that complete infectious viral particle is what we call a virion.
The virion, got it.
At its most basic, a virion consists of two primary components. A nucleic acid core, which is its genome, the virus's genetic instruction manual.
The blueprint.
Exactly. Surrounded by a protective protein coat called a capsid. Together, these two elements form what's known as the nucleocapsid.
Nucleocapsid, nucleic acid plus capsid. Makes sense.
Precisely. Importantly, some viruses then have an additional outermost layer and envelope. But not all viruses possess this. We'll get into that distinction.
Okay, so the genome is the brains, the genetic core, with all the instructions for making more viruses. And we've already established that it's always either DNA or RNA, never both. That's a fundamental defining characteristic right there.
Precisely. That's a hard and fast rule in virology.
Yeah.
And even within that DNA or RNA constraint, there's immense variety in how that genetic material is structured.
How so?
Well, the nucleic acid can be single-stranded, SSS, or double-stranded, DS. It can be linear, like a thread, or in some cases circular, like a loop. What's particularly fascinating is the case of some RNA viruses, like the influenza virus, where their genome is segmented.
Segmented, meaning it's in pieces.
Exactly. It's broken up into several separate molecules, each typically encoding an individual gene or maybe a couple of genes. This segmentation has significant implications for how they evolve and exchange genetic information, which we'll definitely touch on later. It allows for easier mixing and matching.
Ah, okay. That sounds important for things like the flu changing every year.
It absolutely is.
Yeah.
We also classify RNA genomes by their sense. Positive sense RNA can act directly as messenger RNA, ready for immediate translation into proteins by the host cell's ribosomes.
So it's ready to go.
Right. Negative sense RNA, on the other hand, requires an extra transcription step. It has to be converted into a positive sense copy first before it can be translated. It's like needing an adapter.
Okay, positive sense and negative sense. Another layer of complexity.
And the sheer quantity of genetic material can vary wildly, too. From just a few thousand nucleotides letters in the genetic code to hundreds of thousands. Some viruses are genetically very simple, others quite complex.
So the genome holds the intricate instructions. Then there's the capsid, which is like the protective shell you mentioned. What exactly is it made of, and what are its critical jobs?
The capsid is indeed that protective protein shell. It's not a single solid piece. It's composed of numerous individual protein subunits called capsomeres.
Capsomeres, okay.
The specific arrangement and type of these capsomeres are highly characteristic of a particular virus type. Think of them as the specific building blocks for that virus's shell.
And it does more than just protect, right?
Or absolutely. The capsid performs several crucial roles. Firstly, yes, it robustly protects the delicate nucleic acid from physical destruction and enzymatic inactivation, for example, by nucleuses in the environment that would just chew it up.
Got to protect the blueprint.
Exactly. Secondly, and very importantly, it provides the precise binding sites, those viral attachment proteins we'll talk about later, that enable the virus to attach to specific receptor sites on the host cell.
So it's the part that actually recognizes and latches onto the cell.
Correct. Think of it as the specific key that unlocks the cell door, allowing entry. It also facilitates the assembly and packaging of the viral genetic information once inside the host, and it serves as a vehicle for transmission from one host to another.
Okay.
Finally, the capsid is highly antigenic.
Antigenic, meaning?
Meaning our immune system recognizes it as foreign. It plays a crucial role in triggering the host's defense response, which is why our immune system learns to fight specific viruses based on recognizing their capsids, or envelope proteins if they have them.
It's like a sophisticated little armored vehicle with very precise docking stations, and the way those capsomeres are arranged determines the virus's symmetry, which leads to some truly architectural wonders in the microscopic world.
That's a perfect way to put it. We recognize three main types of symmetry in viral capsids based on how those capsomeres come together.
Okay, what are they?
First is icosahedral symmetry. This describes a protein arrangement in the shape of an icosahedron, which is a 20-sided solid with equilateral triangular faces. It's a very efficient way to build a strong closed shell from repeating subunits.
Like a tiny geodesic dome.
Exactly like that. Herpes viruses and adenoviruses are classic examples of this icosahedral structure.
Okay.
Then there's helical symmetry, where the capsomeres surround the nucleic acid in a helix or spiral, forming a sort of tubular nucleocapsid.
Like a spiral staircase around the genetic material.
Precisely. Many mammalian RNA viruses, often enclosed in an envelope, like rabies virus and influenza virus, exhibit this helical structure. The capsid itself is like a flexible tube.
And then for the viruses that are just too complex for those neat geometric categories.
Right, you have complex symmetry. These are the ones that really don't fit the simpler icosahedral or helical patterns due to their intricate and often irregular structures. Think of retroviruses like HIV or the really large pox viruses.
These are definitely the architectural marvels of the viral world. Just amazing complexity on such a small scale.
Absolutely. And speaking of structures, some viruses have that additional outermost layer we mentioned earlier, the envelope.
The envelope, or the lack thereof, is super important, especially when we think about how viruses spread and how we clean surfaces, isn't it? This is a truly practical insight.
It is, very much so. An envelope is an outer lipoprotein sheath that some viruses possess.
Lipoprotein, so fat and protein.
Exactly. And what's unique about it is that it's actually derived from the host cell membrane during viral release, a process called budding. The virus essentially steals a piece of the host cell's membrane on its way out.
Wow, it takes the souvenir.
Kind of. Viruses without this envelope are referred to as naked viruses. The envelope itself is composed of lipids, mostly phospholipids and cholesterol, taken directly from the host cell membrane, and critically, it also contains virus-encoded proteins embedded within or projecting from that lipid layer.
And these viral proteins often appear as spikes on the surface, right?
Yes, exactly. These spikes are called peplomers or sometimes just spikes.
And these poplumers aren't just decorative, are they? They're vital grappling hooks for the virus.
No, they're highly functional, not decorative at all. These spikes often mediate the initial attachment of the virus to host cells, acting as those specific viral attachment proteins we mentioned earlier, the keys to the cell door for enveloped viruses.
Can you give an example?
The really well-known example is the hemagglutinin, HA, and neuraminidase, NA, spikes of the influenza virus.
Ah, H and N in H1N1.
Precisely. Those terms you might recognize from discussions about flu strains and vaccines. These are the very proteins targeted by our immune system and by vaccines, because they are crucial for the virus's ability to infect.
Ugh. So those aren't just scientific jargon. They're describing actual structures that are crucial for the virus's ability to infect. And this brings us to a key why you should care point about envelopes. Their environmental stability, or lack thereof, has real-world implications for how we prevent transmission.
Yes, absolutely. Enveloped viruses are environmentally labile.
Labile, meaning fragile.
Fragile, yes. They are quite easily disrupted by things like acids, detergents, drying, and heat, because that lipid envelope is vulnerable. Consequently, they typically spread through direct contact with body fluids, secretions, respiratory droplets, or through more intimate means like organ transplants. They don't survive well on dry surfaces for long periods.
Okay.
Naked viruses, on the other hand, because they lack this vulnerable lipid envelope, are much more resistant to these harsh environmental conditions. They can often survive longer on surfaces and are sometimes harder to kill with disinfectants.
This distinction is incredibly relevant to our daily lives. It explains why simply washing your hands thoroughly with soap, which is a detergent, is so effective against enveloped viruses like influenza or coronaviruses, because it literally breaks down that fatty outer envelope, inactivating the virus.
It dissolves the envelope, yes.
But for naked viruses, like some that cause stomach bugs, like norovirus,
A classic example.
basic soap and water might be less effective at inactivating them because they don't have that vulnerable outer layer. Soap still helps remove them physically, but it doesn't destroy them as easily. Often requires stronger disinfectants or physical scrubbing.
It's a critical difference in their vulnerability and, consequently, in our strategies for controlling their spread. Hand washing is always good, but the mechanism of inactivation is different.
Good point.
And while viruses generally rely on the host's machinery, some do bring along their own specialized tools to help with the takeover.
Like little toolkits.
Sort of. For instance, in viruses like herpes viruses, there's an amorphous structure called the tegument located between the envelope and the capsid. Yes, and it contains various viral proteins and enzymes that get injected into the cell upon entry, ready to go to work immediately, even before the virus starts making new proteins.
Sneaky.
And some viruses, despite being metabolically inert outside a host cell, carry their own essential or accessory enzymes within the variant itself.
Like HIV's reverse transcriptase.
That's the prime example. HIV is an RNA virus, but it needs to make a DNA copy of its genome to integrate into the host chromosome. It carries its own reverse transcriptase enzyme to do this crucial step.
Which is absolutely crucial for its replication process, allowing it to convert its RNA genome into DNA within the host cell.
Exactly. Or think about neuraminidase in influenza, which helps the virus release from the host cell, or lysozyme in some bacteriophages, which helps them break through the bacterial cell wall.
So this just goes to show that even within their elegant simplicity, viruses have these hidden helpers, these specialized tools that allow them to be incredibly efficient at what they do, hijacking cells.
It highlights their evolutionary ingenuity. They bring only what they absolutely need and can't reliably get from the host.
Okay, so we know what viruses are, and we've peered inside their intricate structures. Now, let's talk about the main event, how they actually make more of themselves. It's not as simple as just splitting in two like bacteria. It's a true biological takeover, isn't it?
It is indeed a highly complex and orchestrated process, often referred to as the viral life cycle. Although, as we said, it's certainly not life in the traditional sense.
Right, more like a replication cycle.
Exactly. While the exact mechanisms can vary greatly among different virus families, the core concept remains their complete dependence on the host cell's synthetic machinery. It's the ultimate biological hijack, where the virus essentially rewrites the host cell's operating system to serve its own replication.
So how does this cellular hostage situation begin? What's the crucial first step in this elaborate viral replication cycle?
The first crucial step is called absorption, or simply attachment.
Okay, latching on.
Yes. The virus must first physically attach to specific complementary receptor sites found on the surface of the host cell. This binding is mediated by viral attachment proteins, or VAPs,
VAPs.
which are part of the virion structure often located on the capsid for naked viruses, or on those peplomers, the spikes, if an envelope is present.
And it has to be specific, right? Like a key in a lock.
Think of it exactly like a specific key fitting into a specific log on the host cell surface. Without that perfect molecular match, infection cannot proceed. This specificity is why viruses often only infect certain cell types, like respiratory cells for the flu or certain species.
So once the virus has its key in the lock, how does it actually get inside the cell? How does it cross that cellular barrier?
That's the penetration or uptake stage. There are a few different methods for this, depending on the virus.
Okay.
For non-enveloped viruses and some enveloped ones, they might enter via endocytosis.
Like the cell swallowing it.
Essentially, yes. The host cell membrane engulfs the virus in a small vesicle, bringing it inside. It's a normal cellular process that the virus exploits.
Clever.
Enveloped viruses often employ direct fusion of their viral envelope with the host cell's plasma membrane.
Merging.
Yes, their envelope literally fuses with the cell membrane, effectively merging the two and releasing the virus's internal contents, the nucleocapsid, directly into the cell's cytoplasm.
Wow. Just slips right in.
And in some rarer cases, there can be direct translocation across the membrane, where the virus or just its genome somehow passes directly through the cell's outer boundary, though this is less common.
Okay. Once it's inside the cell, the virus isn't immediately ready to start replicating, right? There's an unboxing process that needs to happen.
Yes, exactly. That's uncoding, sometimes also referred to as the start of the eclipse phase. This is where the viral nucleic acid, the genome, is separated from its protective protein coat, the capsid.
Got to release the instructions.
Right. Once uncoded, the viral genome is finally free to act as a template for messenger RNA synthesis, or in some cases, directly as mRNA, which is the very first step towards replication. This uncoding process often involves enzymes from the host cell's lysosomes, effectively using the host's own cellular recycling machinery to disassemble the viral particle.
Using the cell against itself again.
It's a common theme.
And after uncoding, the virus really takes over. This is the manufacturing phase, where the cell is forced to stop making its own components and instead starts churning out viral parts.
That's the biosynthesis stage. This is a highly active period involving three key processes, all leveraging the host cell's machinery. First, the transcription of viral mRNA from the viral genome.
Making the message.
Second, the translation of that mRNA into viral proteins by the host ribosomes. These include both structural proteins that will make up new virions and non-structural enzymes that are crucial for the replication process itself.
Building the parts and the tools.
And finally, the replication of the viral nucleic acid itself, making copies of the viral genome.
Okay, and where does all this happen?
The location for this intense activity varies. DNA viruses typically synthesize their DNA and assemble in the host cell's nucleus using the host's DNA replication machinery.
Makes sense.
With poxviruses being a notable exception, as they replicate entirely in the cytoplasm, bringing their own machinery.
Ah, the complex ones.
Most RNA viruses, conversely, multiply entirely in the cytoplasm using RNA-dependent RNA polymerases, which are often viral enzymes, because host cells don't usually copy RNA from RNA.
Okay.
Though orthomyxoviruses like influenza and retroviruses like HIV are exceptions that have steps involving the nucleus.
So the cell's machinery is now completely reprogrammed to be a viral factory. Once all the parts, genomes, proteins are made, they need to be put together like an assembly line.
Correct. That's maturation or assembly. This is where the newly synthesized viral components, the nucleic acids and the proteins, are precisely assembled into progeny virions, or new complete viral particles.
That sounds like a delicate process.
It's incredibly intricate. It often happens spontaneously, driven by the interactions between the proteins and nucleic acids, but sometimes involves helper proteins. This assembly can occur in various locations within the cell, depending on the virus, in the nucleus, the cytoplasm, or even at the plasma membrane just before release. It's a highly efficient process, ensuring the genetic material is correctly packaged within its protective coat.
And then the grand finale, the newly formed virions have to escape to go infect new cells and continue the cycle. How do they get out?
The final step is release. The method of release often depends on whether the virus is enveloped or non-enveloped.
Back to the envelope difference.
Exactly. Enveloped viruses frequently bud gradually from the host cell membrane, or sometimes internal membranes like the Golgi or ER. As they push through the membrane, they acquire their lipid envelopes studded with those viral spike proteins. This budding process may allow the host cell to survive, at least for a while, acting as a continuous viral factory, shedding viruses over time.
A slow leak, almost.
Kind of. Non-enveloped viruses, however, typically cause cell lysis.
Lysis meaning bursting.
Yes, they literally rupture and destroy the host cell to escape, releasing all the progeny virions at once. This is often a much more destructive exit for the host cell, causing rapid tissue damage.
That's a pretty violent end for the cell in many cases. Now, you mentioned something earlier, the eclipse phase. Can you expand on that unique period during replication? It sounds like a moment of viral invisibility.
The eclipse phase is a truly fascinating concept in virology. It's defined as the period where, even though a cell is infected and viral replication is actively occurring,
Right.
no intact infectious virus particles can be detected inside the host cell if you were to break it open and look.
So the virus disappears.
Functionally, yes, as an
Okay.
infectious unit. It lasts from the moment of penetration and uncoating until the first appearance of mature, fully assembled daughter virions during the maturation stage. During this time, the virus is uncoated, its genome is being replicated, its proteins are being synthesized, but it hasn't yet reassembled into an infectious particle.
So it's essentially hidden in pieces, unquantifiable as a complete virus.
Exactly. This makes it a challenging period to target with antiviral treatments because there's no complete virion structure to attack, and it also makes it harder for the host's early immune responses to detect what's happening inside the cell.
So even if a cell is technically infected and making viral parts, there's this window where the virus is invisible, still in pieces, before it becomes a fully formed infectious particle again. That's quite a clever trick. But what happens when the virus's replication cycle isn't perfect, when things go wrong? For the virus anyway. Does that happen often?
Oh yes, even these incredibly efficient hijackers can have hiccups or deviations from the perfect plan. Replication isn't always 100% efficient. We see what are called abnormal replicative cycles.
Like what?
For example, you can get incomplete viruses. These can result from defective assembly processes, maybe a shortage of one component, leading to non-infected virions. The influenza virus, for instance, under certain conditions, can produce a lot of particles that have the envelope and spikes, but lack the full genome, making them non-infectious.
So the virus looks like it's there, maybe triggers an immune response, but it can't actually do its job. It's like building a car without an engine.
Exactly. Then there are abortive infections, where viruses simply fail to produce any infectious progeny. This might happen because the host cell isn't permissive for that particular virus. Maybe it lacks a necessary receptor or an internal factor the virus needs, or the virus itself is somehow defective and can't complete the cycle. The mission fails.
Okay, so the infection just fizzles out.
Right. A particularly interesting type is defective viruses. These are genetically deficient viruses that absolutely require a helper virus to provide missing functions for their replication.
So one virus needs another virus to even exist and replicate. That's a truly surprising aha moment for how different viruses can interact.
It is. The classic example is hepatitis D virus. It's defective. It can't make its own envelope protein. It absolutely needs hepatitis B virus to be present in the same cell to provide the envelope proteins so hepatitis D can assemble and spread.
So hepatitis D effectively parasitizes hepatitis B. That's wild.
It's a clear example of interviral dependency. And finally, you have pseudovirions, a curious anomaly. This happens when viruses accidentally enclose host cell nucleic acid instead of their own viral nucleic acid during assembly.
So it's a virus particle with the wrong instructions inside?
Precisely. It looks like a virion on the outside, might even attach to another cell, but because it doesn't contain the viral genome, it's completely non-infective. It's functionally entered, a mispackaged product at the end of the viral assembly line.
It just goes to show that even these tiny, simple entities are capable of such complex, often destructive, and sometimes even imperfect interactions with our cells. It's not always a flawless process.
No, biology is rarely flawless, even for viruses. But their overall success rate is obviously high enough for them to persist.
Okay, so viruses are these incredible cellular hijackers with intricate life cycles. But they're not static, are they? They are constantly evolving, changing, adapting. This makes them a moving target for our immune systems and for medical interventions like vaccines and drugs. This constant change is key to understanding things like the annual flu shot or antibiotic resistance. Well, antiviral resistance, I mean.
You're absolutely right. That's a crucial point. Viruses are masters of evolution in action, constantly adapting and changing. The primary way they change is through mutation.
Just random errors in copying their genetic material.
Exactly. These are random heritable changes in their viral genomes, and they occur with surprising frequency, especially in RNA viruses.
Why, especially RNA viruses?
Because the enzymes that replicate RNA genomes, the RNA-dependent RNA polymerases, tend to lack the proofreading mechanisms that DNA polymerases have. They make more mistakes, essentially.
Ah, less quality control.
Right. This constant mutation leads to new strains with altered antigenicity, meaning our immune system might not recognize them as easily from a previous infection or vaccination.
Like the flu variants.
Precisely. Or mutations can lead to altered pathogenicity, making them more or less virulent, or even leading to drug resistance, where antiviral drugs no longer work effectively. This is precisely why flu vaccines have to be updated every year, because the circulating strains mutate so rapidly, drifting away from what the vaccine targets.
So it's a constant game of catch-up. And if mutation is like a tiny typo in the instruction manual, then recombination sounds like a full-on gene swap, allowing for bigger jumps in evolution.
It is. Recombination is the exchange of genetic material between two different but related viruses when they happen to co-infect the same cell.
So two viruses in one cell can swap pieces.
Yes. Imagine two different viral strains entering one host cell. During replication, their genomes can get mixed up, and segments can be exchanged, creating a hybrid virus with a new combination of genes. This allows viruses to quickly acquire new traits or adapt to new hosts much faster than through gradual mutation alone. It's a powerful driver of viral evolution, sometimes leading to entirely new, more concerning strains, like pandemic influenza strains that jump from animals to humans.
That's like microscopic gene swapping, allowing them to quickly acquire new traits. Super clever from the virus's perspective, but also super concerning for us. Are there other ways they can mix and match their genetic properties or appearances?
Indeed. We also see what's called phenotypic mixing. In this case, the proteins from different viral strains that co-infect the same cell get phenotypically mixed.
Meaning the outside looks like a mix?
Right. The outer characteristics of the resulting virion might be a blend, maybe using capsid proteins from one virus and envelope proteins from another.
Yeah.
But crucially, the underlying genome inside each virus particle remains original. It hasn't actually changed genetically.
Okay.
A specific case of this is transcapsidation, where one virus's nucleic acid gets accidentally surrounded by a completely different virus's capsid during assembly in a co-infected cell.
So the packaging is different, but the core genetic instruction set is still the original virus's. It might affect which cell it can enter initially, but not its long-term identity.
Exactly. The disguise is temporary. Then there's genotypic mixing, sometimes referred to as polyploidy. This occurs when several nucleocapsids, potentially with slightly different genomes, are enclosed within a single envelope.
Multiple genomes in one particle.
Yes. This is famously seen in double-bold retroviruses like HIV, which always package two copies of their RNA genome. But it can happen in other cases too, where multiple genomes, maybe different variants, are incorporated into one virion without true recombination having occurred.
Interesting. And sometimes viruses can even help each other out, surprisingly enough. You mentioned defective viruses needing helpers.
Yes, that's complementation. Here, one defective virus provides a gene product, a protein, maybe that another co-infecting virus lacks due to its own mutation. This allows the second virus to replicate successfully. But crucially, this doesn't alter their underlying genotypes. The helper virus simply provides a missing function temporarily, acting as a temporary loan of a necessary tool.
So it's like lending a cup of sugar to your neighbor, but for viruses. It's a surprisingly cooperative behavior for such notorious hijackers.
It can be, functionally speaking.
Yeah.
But they can also be competitive.
How so?
That's interference. This is when a mixed infection with two viruses leads to the inhibition of multiplication of one or both of them. One virus gets in the way of the other, maybe by using up resources faster or by triggering an immune response that blocks both. It's an example of viral competition within the cellular environment.
Dog-eat-dog world, even for viruses. Sometimes. Conversely, there's also enhancement. This is where a mixed infection can sometimes increase the overall virus yield or amplify the cytopathic effect, the damage in the host cell. Sometimes for the virus, two really are better than one, perhaps by overwhelming the cell's defenses more effectively together.
So whether through random mutations, gene swapping, temporary disguises, or even accidental cooperation and competition, viruses are constantly adapting and evolving. It's a dynamic, ever-changing foe, making our fight against them a perpetual challenge.
Absolutely. Their ability to change is fundamental to their success.
Given how cunning viruses are at replication and evolution, let's talk about their weaknesses and strengths when faced with the outside world or with our attempts to get rid of them. How do they react to things like heat, disinfectants, etc.? This has massive implications for public health and everyday hygiene. Right. While viruses are incredibly adaptable biologically, they do have physical and chemical vulnerabilities dictated by their structure and composition. It's interesting, generally, due to their simpler structure, lacking complex cell walls or metabolic enzymes compared to bacteria viruses are actually more resistant to many common chemical disinfectants than bacteria are.
More resistant? That seems counterintuitive.
It does, but think about it. Many disinfectants target complex bacterial structures or metabolic pathways that viruses simply don't have. However, their specific physical and chemical properties do dictate what does affect them effectively.
Okay, so let's talk about temperature. We know heat helps sanitize, but what about cold? How do they handle extremes?
Most viruses are quite heat labile.
Labile meaning unstable.
Unstable, yes. They are quickly inactivated at relatively moderate temperatures, like 56 degrees C or 60 degrees C, for about 30 minutes. Think about pasteurization for milk. That process is designed to inactivate microbes, including many viruses, without drastically changing the product.
Okay, so heat is generally bad for them.
Generally, yes. On the flip side, they are remarkably stable at low temperatures, which is why laboratories store viruses for long periods at negative 70 degrees C, or even in liquid nitrogen, or by freeze-drying them. Cold preserves them very well.
Good for storage, bad if you want to kill them with cold.
Exactly. Now, a critical distinction here, related to what we discussed earlier, is that enveloped viruses are particularly heat labile, because their fatty outer layer is more susceptible to disruption by heat compared to a naked protein capsid.
Ah, that envelope makes them more fragile to heat too.
Correct. However, there are always exceptions. Hepatitis B virus, for instance, is unusually resistant to heating. It can survive 60 degrees C for 60 minutes. And prions, which are infectious protein particles, not true viruses, are notoriously resistant even to autoclaving, which is intense heat and pressure.
So not all viruses are created equal when it comes to heat. What about pH? How acidic or alkaline the environment is, does that affect them?
Yes, pH matters. Generally, viruses are stable within a fairly broad pH range, typically between pH 5 and 9. But again, there are specific vulnerabilities linked to their structure or where they infect.
Look.
Rhinoviruses, common causes of the cold, are susceptible to acidic pH. They don't like acid. This makes sense, perhaps, as they primarily infect the upper respiratory tract, not the highly acidic stomach.
Right.
In contrast, enteroviruses, which often cause gastrointestinal issues and do pass through the stomach.
Like polio or hand, foot, and mouth disease viruses.
Exactly. They are highly resistant to acidic pH, allowing them to survive the harsh stomach acid environment and go on to infect the intestines. That's a clear evolutionary adaptation for their preferred infection route.
Makes sense. What about salts? Can they actually protect viruses? That sounds odd.
Yes. Surprisingly, high concentrations, molar concentrations of certain salts like magnesium chloride or magnesium sulfate can actually stabilize the capsid structure of some viruses, like poliovirus, making them more resistant to heat inactivation.
Why is that useful?
This property is quite important in the preparation and storage of some live viral vaccines, like the oral polio vaccine. The salt helps keep the vaccine virus stable during manufacturing, transport, and storage, especially in warmer climates where refrigeration might be challenging.
Interesting. And radiation, UV light from the sun, X-rays, are these effective against viruses?
Absolutely. Ultraviolet UV light, particularly short-wavelength UVC, as well as X-rays and other high-energy particles, are effective at inactivating viruses. They primarily work by damaging the viral nucleic acids, the DNA or RNA, causing mutations or breaks that essentially scramble their genetic code, making replication impossible.
So sunlight can actually help disinfect surfaces.
Yes, the UV component of sunlight does have some disinfectant properties, though its effectiveness depends on intensity and duration. It's also why UV lamps are sometimes used for sterilization in labs, healthcare settings, or even in some air purification systems.
Okay. What about the common chemical disinfectants we use around the house or in hospitals? Which ones work best?
Oxidizing agents are generally very potent antivirals. Things like hydrogen peroxide, potassium permanganate, hypochlorites, the active ingredient in bleach, and organic iodine compounds like povidone iodine.
So bleach is good.
Bleach solutions are very effective. Broad-spectrum disinfectants against many viruses, yes. Also, agents like formaldehyde and beta-propiolactone are active virucidal agents, meaning they specifically kill viruses. They're actually used in the preparation of killed viral vaccines to ensure the viruses are completely inactivated and safe before injection.
But are there viruses resistant to common things like chlorine in pools?
Yes, some viruses, notably hepatitis A and poliovirus, are relatively resistant to standard chlorination levels used in drinking water treatment. This is an important consideration for water safety and requires specific treatment protocols to ensure their inactivation. Adenoviruses can also be quite resistant.
Good to know. This all loops back to our discussion about envelopes again, doesn't it? Because lipid solvents are a huge deal for enveloped viruses. This seems like a critical takeaway for everyday life.
This is perhaps one of the most practical insights, yes. Enveloped viruses are highly sensitive to lipid solvents.
Like what, exactly?
Things like ether, chloroform, ethanol, alcohol, detergents including simple soap and bile salts found in our digestive system. These agents literally disrupt and dissolve their lipid-containing outer envelope, which is essential for their infectivity. Once that envelope is compromised, the virus is typically no longer infectious.
And naked viruses.
Naked viruses lacking this lipid envelope are naturally resistant to these solvents. Alcohol-based hand sanitizers might not work as well against some naked viruses like norovirus compared to enveloped ones like influenza.
This is the practical relevance we talked about. It explains why simply washing your hands thoroughly with soap and water,
Soap being a detergent,
is so incredibly effective against enveloped viruses like influenza or coronaviruses. The soap literally breaks down that outer fatty layer. It's not just physically removing them, it's inactivating them.
Precisely. But for naked viruses, like norovirus or rotavirus, which cause widespread stomach illness, soap is less effective at killing the virus itself, though the mechanical action of washing still helps with physical removal. For those, sometimes disinfectants like bleach are needed for surfaces.
So understanding that envelope makes a huge difference in how we approach hygiene and public health. It's not just academic.
Exactly. It's a very direct application of their structural properties to real-world control measures. And just to reiterate a point we made earlier with emphasis, antibiotics,
Or bacteria.
which are specifically designed to target bacterial processes like building cell walls or bacterial ribosomes, are completely ineffective against viruses because of their fundamentally distinct structure and replication mechanisms. Viruses don't have those targets. This property is actually leveraged in the lab. When trying to isolate viruses from a clinical sample that might also contain bacteria, scientists often add antibiotics to the culture medium. The antibiotics kill the bacteria, but leave the viruses unharmed, allowing the viruses to grow in the cell culture without bacterial contamination.
Using the difference to their advantage. It's the ultimate know your enemy principle, and it affects how doctors treat us every day. And speaking of knowing your enemy, with all this diversity, how do scientists bring order to the chaos of the viral world? How are they classified and named? It must have been a mess initially.
It was. Historically, viruses were classified rather haphazardly, often based on the disease symptoms they caused, like hepatitis viruses causing liver inflammation, or the sites from which they were isolated, terms like pneumotropic for lung-affecting viruses or neurotrophic for those affecting the nervous system, or even just named after the place they were discovered.
Not very systematic.
Not at all. It was quite unscientific and inconsistent. But since 1966, the International Committee on the Taxonomy of Viruses, or ICTV, has introduced and continuously refines a systematic, hierarchical, globally accepted approach based on the virus's inherent properties.
Okay, so it's like a scientific family tree for viruses based on specific measurable characteristics. What are those key classification attributes that scientists use to group them into families, genera, and species?
The modern classification system relies on a combination of fundamental characteristics. The most important are, first, the type of nucleic acid they contain. Is it DNA or RNA?
The basic split.
Right. Second, is that nucleic acid single-stranded or double-stranded? And for single-stranded RNA, is it positive sense or negative sense?
Okay, genome details.
Third, their means of replication, the overall strategy they use to copy their genome and make proteins. Fourth, their morphology, what do they look like? This includes their overall size, distinctive shape, and the symmetry of their capsid, icosahedral, helical, or complex.
Structure matters.
Yes. Fifth, the presence or absence of that outer membrane envelope. That's a major dividing line.
Enveloped or naked.
Exactly. Using these core attributes, the ICTV groups viruses into orders, families, subfamilies, genera, and species.
That sounds much more organized than just naming them after diseases. And they
Yeah.
even have specific naming conventions, right? Like endings for family names.
Yes, there's a clear nomenclature to make it consistent. Family names consistently end in the suffix viridae, like herpesviridae or coronaviridae. Genera end in virus, like simplex virus or beta coronavirus. And orders, which are a broader classification above families, end in ales. Subfamilies can also be designated with arinae.
Okay. Neyer, virid, Neyer, virus, ales. Got it.
This systematic nomenclature provides a clear, universally understood way to organize and communicate about the vast and ever-growing diversity of viruses, allowing scientists worldwide to speak the same language about these microscopic entities. It reflects their evolutionary relationships much better than the old ways.
Bringing order to the viral zoo. Okay, so we've broken down what viruses are, how they're built, how they invade cells, how they evolve, and how they react to their environment. Now, for the real nitty-gritty practical part, how do scientists actually catch these invisible entities and grow them in a lab? You said it's not like bacteria on a petri dish. This is where scientific ingenuity truly shines.
You're absolutely right. This circles back to our foundational concept. Viruses are obligate intracellular parasites.
They need living cells.
They only replicate within living host cells. This fundamental characteristic completely dictates how we grow them in a laboratory setting. You simply cannot put them on an inanimate nutrient medium like agar in a petri dish, as you would with bacteria or fungi. They need live cells to hijack. This was a huge challenge for early virologists.
So how did scientists first tackle this challenge before our sophisticated modern labs? What were the old school methods for cultivation? If you can't use a dish, what do you use?
One of the earliest methods, going back to the very beginnings of virology, was animal inoculation.
Injecting animals.
Essentially, yes. Researchers would inoculate susceptible laboratory animals like mice, rabbits, guinea pigs, sometimes even monkeys, with material suspected of containing a virus.
What was the goal?
It was primarily used for the initial isolation of certain viruses, especially those that didn't grow easily elsewhere. Also for studying how viral diseases develop their pathogenesis, understanding the immune response, and studying epidemiology. It was also important for early research into oncogenesis, the development of cancer, as some viruses were first linked to cancer through animal studies.
Can you give some examples?
Sure. Early poliovirus research heavily relied on inoculating monkeys. Rabies virus was studied in rabbits and mice. Even today, some research, like studying leprosy, which is caused by a bacterium that can't be grown in culture, uses armadillos or mouse foot pads. For virology, though, animal inoculation was foundational but had major limitations.
What were the drawbacks?
Well, it was, and is, expensive, requires specialized facilities, raises significant ethical concerns about animal welfare, and can pose risks to the researchers handling infected animals. Also, crucially, it often only indicated the presence or absence of infectious viruses did the animal get sick or not. It wasn't very good for accurately quantifying the amount of virus present.
So scientists needed a better solution, one that was more controlled, quantifiable, ethical, and maybe cheaper. And that's where the egg-cellent solution came in, right? Embryonated eggs.
A significant leap forward indeed. The embryonated hen's egg, basically a fertilized developing chick egg, was first successfully used for virus cultivation by Goodpasture in 1931, with further development by Burnett. It offered a sterile, self-contained, readily available living system.
Why eggs?
Developing chick embryos contain several different membranes and cavities, each supporting the growth of different types of viruses. Plus, they lack a fully developed immune system initially, making them more susceptible. Typically, 8 to 11-day-old chick eggs are used, and they can be inoculated via several different routes, depending on the virus being studied.
And different routes allow different viruses to grow in specific ways. That sounds quite ingenious. What were some of these routes, and what could you grow there?
Okay, one key route was inoculation onto the choriolitoic membrane, or CAM.
The CAM.
It's a membrane lining the inside of the eggshell. This was a popular site for cultivating viruses like herpes, simplex virus, vaccinia virus, used for smallpox vaccine, and the actual smallpox virus, variola.
And you could see the virus growing.
Yes. What was particularly useful about the CAM is that some viruses, especially pox viruses, would produce visible lesions called pox on the membrane surface. These look like little opaque spots.
Pox, like chicken pox.
Related concept, yes. Crucially, under optimal conditions, each infectious virus particle landing on the CAM could initiate one POC. This allowed scientists to literally count these POCs to assay, or quantify, the amount of infectious virus present in a sample, a massive step forward in quantitative virology. They could even distinguish viruses based on POC appearance, Smallpox made small white pox. Vaccinia made larger grayish ones.
Wow, counting pox on an egg membrane, that's clever. What other routes?
Another major route was inoculation into the allantoic cavity. This is a fluid-filled sac within the egg. This route was often used for serial passages, growing the virus repeatedly to adapt it or increase its concentration, and especially to obtain large quantities of viruses for vaccine preparation.
Like flu vaccine.
Exactly. Influenza virus grows extremely well in the allantoic cavity, and this method is still used today for producing a large portion of the world's seasonal flu vaccines, also used historically for yellow fever and rabies vaccine strains.
So that was like a viral factory for early vaccines, and even some today.
Precisely. Then there was the amniotic cavity, another fluid-filled sac closer to the embryo, mainly used for the primary isolation of influenza virus. And finally, the yolk sac, which is rich in nutrients, was used for cultivating some viruses like Japanese encephalitis and West Nile virus, as well as some bacteria-like organisms such as chlamydia and rickettsia.
It sounds like a fantastic and versatile method, providing so much more control and precision than animal inoculation, but you said it's largely been replaced now for routine diagnosis.
Yes, for routine virus isolation and diagnosis in clinical virology laboratories today, chick embryo inoculation has largely been replaced by cell cultures, although it remains vital for specific research and vaccine production, particularly influenza vaccine.
Which brings us to the modern standard approach, cell culture, also known as tissue culture. This is the mainstay of diagnostic virology today, isn't it? This seems like where the real ingenuity of modern lab work comes into play, growing cells outside the body.
It absolutely is the most widely used system now, forming the backbone of much of our viral research, vaccine development, and clinical diagnosis. The basic process involves taking tissues from animals or humans and dissociating them into their individual component cells, usually using gentle proteolytic enzymes like trypsin that break down the connections between cells.
Okay, so you get a suspension of single cells.
Right, and then you suspend these cells in a carefully prepared, sterile growth medium. This liquid medium contains essential nutrients like amino acids and vitamins, salts, glucose for energy, often supplemented with fetal calf serum, which provides crucial growth factors, and usually includes antibiotics and antifungals to prevent bacterial or fungal contamination.
Like a soup for cells.
A very specific, nutritious soup. These cells are then dispensed into sterile containers, flasks, petri dishes, tubes, where they typically adhere to the plastic or glass surfaces and begin to grow and divide, eventually forming a confluent monolayer, which is essentially a single continuous sheet of cells covering the surface.
A carpet of cells.
Exactly. And again, the core principle holds true. Viruses cannot grow on the inanimate plastic or in the medium alone. They absolutely require these live cells for growth and replication.
So we're essentially creating a miniature controlled environment with live cells for the viruses to do their thing, but without the complexity and ethical issues of a whole animal or even an egg. Did this method evolve too? Were there earlier versions?
Yes, the methods for this have also evolved. Early attempts included organ culture, where small pieces of organs were maintained in vitro, trying to preserve their original structure and function. Historically, this was used for isolating viruses with very specific tissue affinities, like trying to grow certain coronaviruses in tracheal ring organ culture. However, it's technically difficult, labor-intensive, and rarely used now. Similarly, explant culture, where minced tissue fragments rather than dissociated cells were placed in a dish and cells grew out from the fragments, was used sometimes. For instance, adenoid tissue explant cultures were used for isolating adenoviruses. But again, it's seldom employed today because monolayer cultures are much more convenient and standardized.
So scientists realized it was more efficient to just grow the dissociated cells themselves rather than whole organs or fragments. And that leads us to the different types of cell culture or monolayer culture that are the workhorses today. What are the main types?
There are basically three main categories. First, you have primary cell cultures. These are derived directly from animal or human tissues for each use. For example, monkey kidney cells or chick embryo fibroblasts. They are essentially normal cells taken straight from the organism.
And they behave like normal cells.
Yes, but they have a finite lifespan in culture. They can only divide a certain number of times, maybe 5 to 10 passages, where you split the culture before they naturally senesce and die. So you have to constantly prepare new ones from fresh tissue.
Okay, limited use. What's next?
Next are diploid cell strains. These are typically derived from embryonic tissues, like human embryonic lung cell strains. Famous examples are WI38 and MRC5. These cells are diploid, meaning they have the normal number of chromosomes, but they can undergo a much larger, though still limited, number of passages, perhaps 50 or more.
So they last longer.
Yes, considerably longer than primary cells. This makes them very useful, especially for producing human vaccines, as they are well characterized and considered safe.
And finally, the superstars of the lab, the continuous cell lines. These are like the immortalized cells of virology, aren't they? They just keep going.
They are indeed the immortalized workhorses. Continuous cell lines are cells that, usually due to spontaneous mutation or sometimes deliberate modification, have acquired the ability to be propagated indefinitely in culture. They escape normal senescence.
Like cancer cells.
Often they are derived from tumors, like the famous HeLa cell line from cervical cancer, or they've undergone transformation in culture. Other common examples include HEP2 from a human laryngeal carcinoma, BHK21, baby hamster kidney, Vero, African green monkey kidney, and KB cells.
And why are they so valuable?
Because they provide a consistent, relatively easy-to-grow, inexhaustible source of cells. This makes them incredibly valuable for large-scale virus production, like for some vaccine manufacture, for routine diagnostic testing where you need reliable cells day after day, and for basic research. They truly are the workhorse cells of modern virology labs, allowing for standardized, repeatable experiments.
Okay, so once you have these cell cultures, primary, diploid, or continuous, how do you actually infect them with a sample, like from a patient, and then know if the virus is growing? Because, of course, you can't see the virus itself directly with a standard microscope.
Right. After preparing the cell monolayer, you inoculate it with a clinical sample, maybe a throat swab, spinal fluid, stool sample, treated to remove bacteria. Then the cultures are incubated, often in a controlled environment with specific temperature and sometimes with 10% carbon dioxide, which can enhance the growth of some viruses.
And then you just watch.
Essentially, yes. You examine the cultures microscopically every day, usually for about 10 days to 2 weeks, looking for visible evidence of viral growth. And that evidence comes in the form of something called the cytopathic effect, or CPE.
Ah, CPE. This is how scientists actually see the invisible virus at work, isn't it? It's the virus's footprint on the cell, the telltale signs that the hijack is underway and causing damage.
Exactly. The cytopathic effect, or CPE, refers to the adverse cellular consequences of viral infection that become visible when you look at the virus-infected cells under a microscope, comparing them to uninfected control cells. Many viruses produce very characteristic morphological changes, changes in shape and structure in the cells they infect. Observing these changes is a crucial first step in identifying the virus, helping scientists narrow down the possibilities.
So what's actually happening at the cellular level to cause these visible changes? How are viruses damaging the cells to produce CPE? What are the mechanisms?
The cellular changes that lead to CPE can result from several underlying mechanisms, often working together. Primarily, it's often due to the viral takeover of the cell's macromolecular synthesis machinery. The virus forces the cell to stop making its own essential proteins in DNA and RNA and make viral components instead, which disrupts normal function.
The hijack itself causes damage.
Yes. Secondly, it can be caused by the sheer accumulation of viral proteins, or complete viral particles within the cell. These can aggregate, forming those inclusion bodies we'll talk about, or physically disrupt the cellular architecture, like the cytoskeleton.
Just clogging up the works.
Pretty much. And finally, it involves the direct alteration or disruption of cellular structures or functions. For example, some viral proteins can damage cell membranes or interfere with energy production, leading to cell dysfunction and death.
So it's essentially the cell breaking down or changing shape as the virus takes over and multiplies. And there are distinct observable types of this cellular mayhem that scientists look for, each like a unique signature for different viruses. What's the most common, most dramatic one?
One of the most common and dramatic forms of CPE is cell death, cytocidal effect, and lysis.
Just killing the cell outright.
Yes. This is where the virus replicates so aggressively that it causes the host cells to die quickly and often rupture, or lyse. Under the microscope, you'd observe the cells rounding up, shrinking, crenation, detaching from the surface, and eventually the entire monolayer sheet of cells disintegrating. Viruses like polioviruses, herpes simplex virus, toga viruses, and pox viruses are classic examples that cause this kind of extensive, destructive CPE.
Ouch. A complete destruction of the cell carpet. But other viruses might just make the cells look a bit sick or change shape without completely destroying them right away.
That's right. Another common type is rounding of cells and degeneration. Viral replication can lead to changes like nuclear pinosis. That's when the cell's nucleus condenses and shrivels up, becoming dense and dark. This is often accompanied by the cells rounding up, losing their normal shape, becoming more refractile, brighter under certain microscopy, and generally degenerating, eventually detaching. Picornaviruses, the family that includes common cold rhinoviruses and enteroviruses, often show this rounding effect. Herpes simplex virus can also cause discrete focal degeneration, like little patches of damage that spread outwards.
Okay, rounding and shrinking. And then there's a particularly fascinating one you mentioned earlier, syncytia formation, or giant cell formation. This sounds like something out of a science fiction movie, these cells merging together.
It's quite striking to observe visually. Certain viruses cause the membranes of infected cells to fuse with neighboring cells, either infected or uninfected. This results in the formation of large, multinucleated giant cells called syncytia, syn-SIC.
Which viruses do this?
It's characteristic of paramyxoviruses like measles virus and respiratory syncytial virus, RSV, hence the name, but also seen with herpes viruses and human immunodeficiency virus, HIV.
Why do they do that? Is there an advantage?
It's thought that this can facilitate cell-to-cell spread of the virus directly, without the virus having to exit the cell and risk exposure to antibodies or other immune defenses in the extracellular environment. It's a clever viral evasion tactic, essentially creating tunnels for the virus to move through.
That's a truly ingenious way for the virus to spread undetected by the immune system by making its own bridges between cells. Another visible sign of infection involves inclusion bodies. What are those exactly? Are they related to the syncytia?
Not directly related to syncytia, but they are another visible sign of viral activity inside the cell. Inclusion bodies are basically accumulations of viral components like viral proteins, replicated nucleic acids, or even assembled or partially assembled virus particles that form characteristic structures or alterations either in the cytoplasm or in the nucleus of infected cells.
So they're like little viral factories or maybe storage depots or even waste dumps that the virus leaves behind, and you can see them under the microscope.
That's a good way to think about it. Their location, nuclear versus cytoplasmic, size, shape, and how they stain with specific dyes can be quite characteristic of the virus type, acting like internal fingerprints.
Can you give some examples? Where would you find them?
Certainly, intranuclear inclusions found inside the nucleus are typically seen with DNA viruses that assemble in the nucleus, such as herpes viruses, adenoviruses, parvoviruses, and papoviruses. There are even subtypes like Cowdrey type A inclusions seen in herpes or yellow fever, which are large and eosinophilic, pink staining, often pushing the chromatin to the edge of the nucleus. Cowdrey type B are different, seen with adenovirus or poliovirus.
Okay, nucleus inclusions for DNA viruses mostly. What about the cytoplasm?
Intracytoplasmic inclusions are found in the cell's cytoplasm. The classic example is the Negri body, a specific type of eosinophilic inclusion found in the cytoplasm of nerve cells infected with rabies virus. Finding Negri bodies is diagnostic for rabies.
Wow, a specific diagnostic sign. Do any viruses make both?
Yes. Some viruses, like measles virus and human cytomegalovirus, HCMV, uniquely produce both intranuclear and intracytoplasmic inclusions in the same infected cell, providing another very distinctive clue for identification. Pox viruses, like variola, smallpox, produce large cytoplasmic inclusions called Guarnieri bodies, which are basically aggregations of virus particles.
That specificity must be incredibly helpful for diagnosis, like solving a microscopic puzzle. And then there's a type of CPE that connects viruses to something far more sinister, transformation. This is where viruses intersect with the study of cancer, right? A truly surprising connection for many.
That's a profound connection indeed, and cell culture was crucial for discovering it. Certain viruses, known as oncogenic viruses, have the potential to cause cancer. In cell culture, they can induce transformation.
What does transformation look like in a dish?
Transformed cells often lose their normal growth controls. They lose contact inhibition, meaning they don't stop dividing when they touch each other like normal cells do in a monolayer. Instead, they continue to grow, piling up on top of each other, forming dense clusters or foci that look like microscopic tumors on the surface of the culture dish. They may also change shape, becoming more rounded, and can sometimes be grown indefinitely like continuous cell lines.
Which viruses can do this?
Examples include certain herpes viruses, like Epstein-Barr virus, some adenoviruses, hepatinoviruses like hepatitis B, papoviruses like HBV, and retroviruses like HTLV1. Observing this transformation in the lab provides strong evidence for their potential role in causing cancer in a living organism. It's a critical area of ongoing research.
That's a truly surprising and often alarming connection for many people, revealing the deep and sometimes long-term impact viruses can have. Okay, what about something called hemadsorption? Sounds related to blood.
It is. Hemadsorption means adsorption of red blood cells. It's another clever visual trick used for detecting certain viruses, especially those that mature by budding from the cell surface.
How does it work?
For viruses like influenza and parainfluenza viruses, their viral glycoproteins, those spike proteins like hemagglutinin, get inserted into the host cell's plasma membrane before the virus buds out. If you then add erythrocytes, or red blood cells, from certain species, like guinea pig erythrocytes for flu, to the infected cell culture,
What happens?
these red blood cells will stick or adsorb onto the surface of the infected cells because the viral hemagglutinin protein binds to receptors on the red blood cells. So you see clumps of red blood cells stuck to the infected cells under the microscope.
Why is that useful?
This phenomenon indicates viral multiplication, specifically the production of those viral surface proteins. And it's particularly useful because heme adsorption can often occur before other visible CPEs, like cell rounding or detachment, become apparent. It provides an early diagnostic clue that the virus is present and replicating.
So it's an early warning sign that the virus is multiplying, even before you see the cells themselves looking obviously sick. What about metabolic inhibition? How does that show viral presence?
This is a more indirect method. When viruses grow vigorously in cell cultures, they often shut down or significantly inhibit the host cell's normal metabolism to divert resources to viral replication. Healthy cells normally metabolize nutrients in the growth medium and produce acidic byproducts, which lowers the pH of the medium.
Okay.
This pH change is usually visible because the growth medium contains a pH indicator, typically phenol red, which is red at neutral pH, but turns yellow-orange as it becomes acidic. If a virus is growing well and inhibiting cell metabolism, the cells won't produce acid and the medium will stay red, while the medium in uninfected control cultures turns yellow.
So no color change means virus might be present. Clever.
Exactly. It indicates metabolic shutdown. And finally, related to viral interactions we discussed earlier, there's interference.
Where one virus blocks another.
Precisely. The multiplication of one virus in a cell culture can sometimes inhibit the multiplication of a second, different challenge virus that is added later.
How is that useful for detection?
This property can be very useful for detecting viruses that don't produce any obvious CPE themselves. Rubella virus is a classic example. It often grows in cell cultures without causing visible damage.
So how do you know it's there?
You first inoculate the culture with a sample suspected of containing rubella. Then after a few days, you challenge the culture by adding a second virus that normally produces a very clear CPE, like an enterovirus. If the rubella virus was present in the original sample and grew, it will interfere with the replication of the challenge virus, and you won't see the expected CPE from the enterovirus. The lack of CPE from the second virus indirectly indicates the presence of the first interfering virus.
Wow, using one virus to detect another, that's quite sophisticated. So by meticulously observing all these distinct cytopathic effects, cell death, rounding, giant cells, inclusion bodies, transformation, hemadsorption, metabolic changes, and interference, scientists can piece together which invisible foe they're dealing with. It's truly like forensic pathology for microscopic invaders, deciphering the unique patterns of destruction or change they leave behind.
That's an excellent analogy. The type of cell line that supports the virus's growth, combined with the specific nature, timing, and appearance of the cytopathic effect, are invaluable tools in the complex process of identifying and understanding individual viruses. It allows us to build a comprehensive picture of these tiny but incredibly powerful biological agents.
Wow, what a journey through the microscopic world of viruses. We've really gone from understanding the fundamental, almost paradoxical nature of viruses as these obligate parasites.
Not quite alive, but masters of hijacking life.
Exactly. Through their intricate structures, the capsids, the envelopes, the amazing variety of genomes, and their incredibly clever replication strategies, essentially turning cells into factories for themselves.
Meticulously decoding those processes.
All the way to how scientists have painstakingly learned to cultivate and identify these invisible architects of disease, using everything from animals to eggs to sophisticated cell cultures.
It's a history of incredible scientific ingenuity, really. We've seen how their unique structure, specifically having either DNA or RNA, but never both, fundamentally sets them apart from all other life forms.
And why this fundamental difference means antibiotics designed for bacteria simply don't work against them. That's such a crucial practical insight that impacts how we approach infectious disease treatment every single day.
Absolutely. And we've highlighted the vital distinction between enveloped and naked viruses. That difference explains so much, from how they transmit through the environment to why simple soap and water is so effective against enveloped viruses like flu or coronaviruses, but maybe less so for inactivating naked ones like norovirus.
knowledge that directly impacts our hygiene practices. And then we looked at the ingenious ways scientists developed to actually study them in the lab, moving from animal inoculation and those pock-counting assays on embryonated eggs,
which were breakthroughs in their time,
to the sophisticated cell cultures of today, which allow us to observe those telltale cytopathic effects, the viral footprints, in incredible detail, helping us identify the culprit.
It truly is a testament to scientific creativity, constantly adapting methods to the unique, obligate intracellular nature of viruses, allowing us to understand them, diagnose infections, and develop ways to combat them more effectively.
So as we wrap up this deep dive, here's something to ponder, building on everything we've discussed. Given their incredible adaptability, the way viruses constantly mutate and recombine, always generating new variants,
Their sheer evolutionary speed,
and their absolute reliance on hijacking host machinery, what future challenges and maybe surprising breakthroughs do you think lie ahead in our ongoing dance, this co-evolutionary battle with these microscopic masters of cellular takeover?
that's the big question, isn't it?
Will we ever truly win this dance? Or is it a perpetual evolving relationship where we just keep adapting to each other? What new forms of understanding, or perhaps even entirely new therapeutic approaches maybe exploiting their own dependencies might emerge as we continue to unravel their deepest mysteries?
It certainly feels like an ongoing evolutionary arms race. Every new virus that emerges, every new mechanism we uncover, reshapes our understanding and forces us to innovate our strategies, vaccines, antivirals, diagnostics. The more we learn, the more we realize the depth of their cunning, but also potentially new vulnerabilities to target.
Absolutely. It's a field where there's always more to discover about these simple yet incredibly profound biological entities. Thank you for joining us on this deep dive into the viral world.
My pleasure.
We hope you feel a little bit more well-informed, maybe a little bit less fearful in some ways, and perhaps even a little bit more in awe of the intricate and impactful world of viruses that exists all around us and even within us. Keep learning, keep exploring, and we'll see you on the next deep dive.